I'm a staff scientist in the functional MR facility in mental health. And today I want to talk a little bit about just the underlying physics and math and principles that bring us here and support the work that we do. So I'm going to start off with talking about the very source of the MR signal. So this has its roots in quantum physics. I'm not going to get into any of that really, but just talk about some of the models that kind of are useful, that model that system, that let us think about what we're doing and how we get information out of that. And then I cannot do that here. I don't have, so I don't have control over here to fix that. So can you close?
I think that's just be one that's before.
Okay. All right. That's fine. So then we'll get into what the acronym means, magnetic resonance imaging, and it's actually kind of nice that it logically flows in that order, and we'll show that. From there, we'll show how these basic principles underlie and pin the contrast in the images that we're seeing and that we're acquiring. And then at the end, I'll have some just brief things that you want to think about when you're doing your experiments, when you're setting them up, when you're designing things. So, so without much further ado, let's jump in. So for the source of the signal, let's start with the magnets. Turns out that atomic nuclei have, a few of them have these properties where they can be thought of as like tiny magnets. This was the Nobel Prize for this was awarded back in 1952. And some of the common nuclei that have this property of, being like slightly magnetic or magnetic at the microscopic or quantum scale, like carbon-13, sodium 23, which one of our scientists is doing experiments on right now to see if they can do functional sodium imaging. Phosphorus 31 is also common, at least on the floor. We have a couple of experiments looking at phosphorus spectroscopy, and another scientist is supporting some of those efforts. But does anyone can hazard a guess, either here or what's the species or nuclei that we use most commonly for the imaging work that we do? Right. Yeah. So it is protons in water nuclei, in proteins, in fat, and pretty much every molecule in our body. So again, the reason for this is we want to, have a lot of species, and the reason for that will come very apparent very shortly. So in like me standing in the room here, us sitting in the room here, or you looking at here, unless you're in a magnet, if you look at the distribution of these like tiny microscopic magnets in your body, it's randomly aligned. But it turns out if you expose these species with this property where the nuclei look like tiny magnets to a very strong magnetic field, then they start to line up and align themselves with that main field. Now, it still is the case that about the same amount will align parallel, that will align anti-parallel with the field. But because of the number of nuclei that you have, which is in the billions and trillions, there is a small enough percentage difference that it creates a detectable magnet or magnetization that forms the basis of the signal that we can acquire. So we're trying to depict that here, where these nuclei, they're randomly aligned, but you put enough of them in a strong enough magnetic field, and then you will induce you become a tiny bar magnet in an MRI scanner. And this is similar to the fact that you take a magnet and you put it against a piece of iron or steel that was not magnetic, but then you magnetize that piece of material. So similar effect, but smaller magnitude. Okay, so now we have our magnets. Resonance is where we can use energy now to inject and perturb that system to get some information out of it. We're looking at these microscopic quantum type of objects. And one way of thinking about them is that they're spinning, precessing, rotating, vibrating, but at a characteristic frequency. So the model that's useful for us is we have this strong magnetic field that's aligned along one direction. And not only is the little magnet precessing on its own axis, but it's also precessing around or rotating around that main field. There's a nice video that's still up on YouTube that depicts what's happening here. So if we think that our magnet, or if we say that our magnet is aligned along the Z direction here in this diagram, which is vertically up, and the little bar magnet is represented by the yellow arrow. You can see that that bar magnet is rotating or precessing around the main magnetic field. So when your subject sample is in the MRI scanner, when they're in there and they become these weak magnets in the system, this is what is happening now as part of that magnetization. The molecules or the hydrogen nuclei are now precessing around that main magnetic field. And there is the simple equation that links the frequency at which they precess around that field to, which is the omega zero. B0 represents the strength of that main field. And that gamma is a quantity called the gyromagnetic ratio. And that determines how quickly now a given type of nuclei will precess around the main field, around B0. And for hydrogen protons, that value is approximately 42.5 megahertz per Tesla. So the most common clinical field started off at about 0.5 T. So there, the imaging frequency was 42 divided by 2, which was about 21 megahertz. The most common field after that was 1.5 T, so there you're around 61, 62 megahertz. Most commonly, down in the center, we have 3 Tesla systems. So 42 times 3, that takes you to 126 megahertz. So the fields there, imaging fields, the magnets are ramped and tuned slightly differently. So it's around 125 to 126 megahertz for 3T. And then for 7T, we're right around 300 megahertz for imaging. And we have a few of those systems on the floor as well. So we have our protons in our main magnetic field, and they're precessing around that field parallel, in a generally parallel direction. But in that state, we can't detect them. So even though that magnetization has been present, as long as that magnetization of the sample is parallel to the main field, we can't see it. So what we have to do is put some energy into that system and disturb the alignment and make some or all of that magnetization perpendicular to the main field so we can detect it. And that is where we exploit resonance. And why this happens is, I like this analogy of, one person pushing another on the swing. If you go with the case that's read out on the right side, if the person is swinging and then the person on the swing being pushed and the person pushing don't match up, and the person that's trying to push is doing it randomly, the person on the swing doesn't go very far. Occasionally, they might get some energy from the person pushing, but it's a very inefficient process. It's wasted, and the person on the swing doesn't move very far or very, a lot. Whereas if the person pushing matches exactly when the person on the swing swings back to them and pushes them away at that time, it becomes a much more efficient process. And then the person on the swing moves further. And this is what's happening. This is the analogy of what's happening at the quantum nuclear level. If you're vibrating and precessing, if you have energy being applied, it won't do anything to your alignment unless that energy matches the frequency at which you're precessing. So that's depicted here, where we have that sine wave on the XY plane represents an RF field or a B1 field, and its frequency matches the frequency at which the little nuclei magnet is precessing around the main field. And if those two match, then you can tip that magnetization into the perpendicular XY plane where we can detect it. So this is just recapping we were just saying. So you need to knock or excite your protons out of parallel alignment into perpendicular orientation so we can detect it. You need to be on resonance. You need to match those frequencies to do it efficiently. And if you don't, you're putting energy into your subject or sample and you're heating them up. So this can cause what we call SAR issues. And there, that's wasting energy. It's potentially dangerous to do that. So now we have our magnets, we have resonance, so we can create a signal now. But because our main magnetic field tends to be very uniform, we don't have any way to generate any spatial. We have not talked about how we generate spatial information yet. So let's get into that section. So in our MR systems here shown schematically, the big outer tube is our main strong magnet, our 1.5T, 3T, 7T magnet. And then there's some volume shown in blue here at the center of the system where it's actually 3T or 7T to a very uniform degree. Uniform parts per million is what it's the specs we shoot for. And the length of the arrows here show nominally the strength of the field. So we try to have the field very uniform at the center of the magnet. But if the field all across this volume is uniform, we have no way to encode, like to tell, okay, if we excite something here versus down here, where the signal is coming from. So to get around that, as part of the imaging process, known control distortions are applied to that main field. And this is usually done by applying, it's called linear gradient fields. And what that does, it gives us now a spatial variation in the magnetic field your species are seeing at each location. So at the very bottom of the imaging volume, the species there are seeing a slightly smaller field than the species at the top. So the ones at the bottom are going to precess or rotate a little bit slower than the ones at the top. And if we go back to our imaging equation, our equation that defines our precession frequency, now we originally had a case where we had one field magnitude, B0, so we had one precessional frequency. But now, when we vary things over space, we now have a spatially varying magnetic field, our Bx. So now we have a spatially varying frequency, that's our omega x. And because we tend to have a very strong main field with a superimposed, smaller, linearly varying field, this is a very common formulation in imaging, where omega x is the gyromagnetic ratio, and then the B0, which is the constant main field, and you add on a spatially varying gradient, which is smaller. Any questions about this, or is this generally okay so far? All right. So how do we use this? So the first way, have folks heard of slice selection? So where you want to just image one slab of tissue, one like fairly small, well-defined slab of tissue. So we take this equation and let's vary the field along the head-foot axis of the subject. So now if we have this variation in main frequency, in the magnetic field along the head-foot axis, we say now, okay, if we apply an RF field at omega-3, we're just going to excite and tip the spins in that slab of tissue, up in the patient's brain. And then bands, like the tissue in omega-1 are not, and omega-2, they're not at resonance, so they're not going to be affected by this. They're not going to be excited. They're not going to, tip into that XY plane, so we're not going to see them. And then after we're done detecting the signals from, the omega-3 location, then we can repeat and essentially transmit at each of the frequencies for our respective locations that, that we want to image. And this is one of the first things the scanners do when you prescribe your volume. It looks at what volume you're going to acquire, how thick your slices are, the number of locations, and it actually builds up an internal frequency table. So it knows when I'm going to excite this location, this is the frequency I have to use to excite the protons, usually in that bit of spatial volume. Okay, so that's how slice selection works. And it turns out that we can actually apply this concept of spatially varying our frequencies more generally. And now we're getting into what we call frequency encoding and phase encoding. And this is applying this spatial variation in field to vary the frequencies of our species and using that to encode spatial information. So it's a little bit of an abstract jump here, but if you bear with me, hopefully this will make maybe a little bit more sense as we go through the next few slides. So in the previous slide, we just excited 3 slabs of tissue in the subject. But now, as we are done with our excitation, and we're just now listening to the protons and the signal they're emitting, if we apply a second gradient now along another axis, so here we're going from the left to right of the patient, and we are going from, if we go patient left, lower frequency, to patient right, higher frequency. Again, now protons on the left side of the patient, they're precessing at a slightly lower frequency than the protons on the right side. So this now, we can detect this group of frequencies, and it turns out that there's a very handy mathematical framework that lets us disentangle these spatially. So that's called Fourier analysis. And we leverage that. This is essentially all of a lot of the underlying MR image reconstruction. We have a signal that has this frequency information encoded in something that looks like that GFT, that the second graph there, that represents sine waves A, B, and C, the red, blue, and green waveforms added up together. Right, so A, B, and C represent your frequencies from the different locations that you're seeing. And what we detect in our systems is the signal GFT, or some variation of that. It ends up being a lot more involved in that because we're detecting many more than just three sine waves here. But in principle, this is what we're doing. And then we can use Fourier analysis to go from GFT to this GFF. And then the amplitudes of the ABCs in the lower right graph tells us how much of each of the frequencies was in that signal we're detecting. So what we're seeing here is that in A, this here, it's unfortunate that they also scale it with the frequency, but the amplitude the top to bottom of the A waveform is larger than the B waveform, which is larger than the C waveform. So that's the meaning of the amplitudes in this graph here, the strength or the amplitude of each of the waveforms up here. This is when we detect the signal and we do this transform, this graph here is actually like a 1D representation of our image, like the pixel intensity values that we're seeing in the images that we acquire. Any questions here? Any questions online? So again, we're taking this concept of spatially varying our frequencies, and we're now using that to encode spatial information into our signal via Fourier encoding. And the field developed this essentially the space in which the data are acquired. We refer to it as K-space. And it's essentially this Fourier space in which all these frequencies are kind of adding up and overlapping. And then we take that K-space, we put it through a Fourier analysis, and we get our image space. However, it turns out there's another very interesting way that you can think about this and how it interacts with your imaging hardware and your imaging gradients. So remember, we talked about we have our protons, our resonance for the most part. This diagram on the left here, this is a pulse sequence, and this shows what the scanner is doing to try to image or get a signal from that proton sample. So the first line here shows what's happening with the RF signal, so the radio frequency system where you're injecting energy. So at the very beginning, there is a little oval there that represents playing an RF pulse, essentially injecting an RF pulse at your sample. But that is overlaid with a gradient on the z-axis here. So this is analogous to what I was showing a couple slides ago with the patient with that z gradient along their head foot. And what this is doing is this GZ is selecting a particular portion of tissue that matches the resonant frequency of this pulse. So this is what is happening so this part of the sequence here, or what the scanner is doing, this pulse sequence, this is the slice selection or slab selection region of the pulse sequence. So now that you have your tissue in that particular region excited now and ready for imaging, the rest now in the XY plane, this now represents where you spatially encode that information so that you can get localization information from different parts of in the tissues in different parts of your sample. Okay. Now, it turns out that if you look at the area of the gradients and how these evolve over time, that actually walks you appropriately along in K-space. So the formal mathematical way of saying this is if you look at the time integral up to a certain point of the gradient amplitudes along that point, you will get to where you are in K-space. So for example, if we just look at Y here, if we start at the B point, we haven't had any activity on the X and Y gradients, so those are at KY and KX equals 0. But at this point, we start applying some amplitude in GY. So then we start traveling along the positive GY direction. So that takes you along the KY axis. So that's going from B here up to C. But we're doing it simultaneously in the negative X direction. So now you're going along negative X, KX, and then we wind up in this corner of our Fourier or K-space. At point C, we turn off these gradients. And then we turn on a positive X gradient. And this tends to be now where we acquire, it will read out our data. So now this walks us now back. So there's no further activity along the Y direction. So we stay at that point along KY and we just walk along the KX direction. And as we walk through this space, we are collecting encoded, spatially encoded information. Questions? Okay. When I was first doing this, this was a little, it took me a long time for me to wrap my head around this. And one thing that finally helped this make sense to me was the fact that Fourier analysis starts off with the theory that you can create any waveform from adding up a set of regularly varying signals. So if you have a large number of sines and cosines, if you weight each of those appropriately, you can reconstruct any time signal. Fourier essentially expands that concept to multiple dimensions. So if you have things that vary at different frequencies along a certain axis, if you have enough of them, you can use those to create any 2D, 3D, 4D representation. And that's what this diagram here is trying to show. So along, let's go to the horizontal direction here. Along, going from center to B1, you have something there that's varying at a relatively high rate along the horizontal direction. B2, you're varying vertically and you don't go as far out, so you're not varying as quickly. And for B3, you're going out along a little bit along X, a little bit along Y. So you're varying a little bit more slowly along both directions. So that's where you have this diagonal variation or hatch pattern. But you can add all these up and you get something that looks like this. And if you add all of these up and you add enough of them up, you can get, you can break any image down into these components, and correspondingly, if you encode that spatial information, using this process, you should be able to detect this again. And this is the very generalized Fourier signal equation. So S of t is what we detect, and it ends up being, the Fourier transform of mxy. That is what we actually want to see. That's what we actually want to image. So if we know this and we know the framework, we just take the inverse Fourier transform of the signal we detect, and we get back our image. So I know this is a lot of hand waving and very general steps, but any questions or any thoughts on how to? Okay. Just practically, here's an example using President Lincoln's portrait. If you run that portrait through Fourier transformer, you get something that looks like this. Its magnitude looks like this. And again, this will look and behave just like the signal you would get if you Fourier encode a sample in an MRI signal, in an MR system. The center represents the average intensity or average power in your image, and things around the center represent things that are varying very slowly over space. And as you get further out to the edges of this Fourier space, then you are encoding higher frequency information or finer detail. And that's shown when you take this image and you break it up and you take away different parts of the Fourier space and then try to reconstruct the image from those. So on the left, you take away the center. So then most of the brightness, most of the energy in the image is gone, but the information that encodes the edges is still there. So you can see the edges in the portrait. Conversely, if you take away the edge information, but leave just the center, you keep most of the energy and you have things that vary more slowly over space. So that's why you're getting some blurring here. Now, in order to collect this data efficiently for fMRI, we want to get this spatial information very, very quickly. So conventional imaging does this first bit here where for GZ and RF, you have your slice selection, and then you do a little bit of encoding and usually acquire one line of data. So then you stop at about this point here in the sequence. But for echoplanar, because we want to run faster and we want to try to capture the brain for fMRI much more quickly, we read out a lot of information in one go. And we end up traversing K-space in this type of trajectory. Okay. And I would urge that if you want to get into this a bit more in detail, the references attached both to this section here and this reference from Pascal and Morris. These are really nice intros talking about the math and the physics underlying these in much more depth than I'm able to go into here. So besides using rectilinear gradients to cover this K-space, there are other trajectories that are possible. Another common one was the spiral trajectory. It's another very efficient way of traversing K-space and encoding that information. And this is still used very much in some applications. But for fMRI and EPI, we tend to like to stick to the rectilinear acquisitions. So a couple of things to keep in mind here. How you're sampling in K-space, i.e. when you read from one point to another, that is actually inversely proportional to your field of view. So if you have tiny steps in K-space for sampling, that means you have a large field of view, versus if you have large steps between your samples, you have a small field of view. So that's one thing to keep in mind. Second is that the extent in your K-space, how far you go out, that determines your resolution, i.e. your voxel size. So remember when we were talking about the portrait, if you get stuff near the center of K-space, that encodes very slowly varying information spatially. So that's low resolution information. If you want the high resolution information, you need to go further out in K-space, i.e. cover much more of it. To do this, your gradients need to need to have gradients that are strong, but also being able to switch quickly. And that's the speed in the gradients. For fMRI, this actually happens to be one of the very limiting things about conventional scanners. For subject comfort, we are limited to 200. I've seen some systems now going up to 250 millitesla per meter. And this is one of the reasons why our facility has been looking into local gradient coils, which do not have this limit imposed on them. Okay, so let me explain why this could be useful for us. So as we're acquiring data here, we want to traverse K-space, but it turns out making these jumps from one line to another and changing the direction of walking through K-space tends to be very expensive in time and energy. So on this graph here, this is an idealized graph. These look like they don't cost any time. But on a real system, so just acquired this a couple days ago, this is acquiring a 3mm 3 echo EPI scan. So between the end of the previous image and this one here, that takes about 80 milliseconds. Okay. In a 2-second TR, it's 25 slices. So that's not a lot for what people expect from our systems. So this is a real constraint in terms of what we're able to get out of the systems and, at what resolution we're able to image and how quickly and how much coverage we can get. It turns out that those transitions between reading in one direction and reading in the opposite direction, you see how much time we spend here going from the negative to the positive direction. It's like half of the time of that transition between you're done reading one lobe to the end of reading the next one. So you see that difference here is about 520 microseconds. So it's half a millisecond. But you're spending about half of that, about 200 milliseconds, just switching from reading one direction to another. You have 60 lines of data in your image. That's 12 milliseconds you've just wasted switching gradients. So this is one reason why the body gradients are limiting to us, and we would like very much to get our hands on local gradients so that we can get past these performance limits that are imposed on the systems by the body gradients. So you don't spend this much time switching between your readout directions. So you can be much more efficient with acquiring your data. So how that works is, we have our whole body gradients here. That circle, that imaging volume that we span, tends in clinical systems, tends to be about 48 to 50 centimeters. And over those volumes, we're constrained to these FDA and IEC slew rate limits. What we're doing with local gradients is we're actually really reducing the volume over which we're switching the fields and where we're imaging. And practically, you can see on our 3TV system where we have one of these coils, this is what the system looks like for body imaging. So that's a 60 centimeter bore there, and that's typical for clinical system. A lot of systems have 70, but because of this, we tend not to like those systems because they take a performance hit for that wider gradient. But with the local gradient put in, you can see how much of that imaging volume you lose. So right to left, that's again 60 centimeters. But with the local gradient put in and the RF coil, that's about an 18 centimeter phantom in there. So you're left with about 20 centimeters of real imaging volume. All right, so that's the price you pay. But in return, you get a lot of speed in terms of being able to switch your fields and acquire functional data much more quickly. And I started putting this together. So if you go to my GIST page on GitHub, and then this should be the first one there right now, but so you don't have to copy the hash. But we started tracking, we only have only listed Siemens and GE gradient systems here because those are the systems we have in-house, and comparing both the standard clinical systems and what each of the vendors offered at one time or another for research systems. So you can see the standard clinical systems. We have 3T A and B, line 2, 3TD in the facility is line 3. So these are fairly typical from a few years ago. Slightly newer, there's a Prisma system here at line 8, which is also in the facility, but that belongs to NIDA and NIAAA. The 7T Terra systems have similar gradients to the Prisma, but the local gradient system I showed in the previous photos, we had a prototype shown here in line 6 when we first started looking into this. So this was a loaner system till the final system was built. So that had a 120 millitesla per meter maximum, but it could slew at 1200 millitesla per meter per millisecond versus 200 for the clinical systems. The final system that was designed and delivered for that, and the one that was actually shown, that has a 140 max, so it's slightly stronger than this, but in the trade-off is it slews slightly slower. So these are some of the trade-offs gradient designers make when they are optimizing their systems.
The technical term slew means it's just switching.
Yeah, so the question is like slew rate. What does that mean? It means how fast the fields can switch. And the facility should take possession of the system here indicated in line 12 in blue which is a Magnus 3T from GE in hopefully 15 to 16 months. And that has a maximum gradient amplitude of 300 millitesla per meter, but slew rates of 750, so 3 times what we're currently able to do on our clinical systems. Okay. So hopefully that gives you a little bit of a sense of what's underlying, the imaging process, setting up encoding data and acquiring it. So let's see how that plays out and gives your images the contrast between the tissues that we're seeing. Okay. So let's again go back to this frame of reference where we're looking at magnet along the z-axis. And when we put our sample in that we want to image, we have a magnetization in the tissue that's parallel to that. Okay. Be excited with some RF, tip it down perpendicular to the main field so we can detect it. And then the system, after we take away that RF, wants to go back to its equilibrium state. So that longitudinal magnetization, that M0, wants to grow back to where it was at equilibrium when we first put it in. And it turns out that regrowth back to equilibrium, that's measured by a contrast quantity called T1. And that's illustrated here, where the magnetization is this gray arrow here, which is almost 0 here. The green arrow is the magnetic field that's applied. And you can see as time is going on, that longitudinal magnetization is growing back. Okay. And why this is important is the next time you image that slab of tissue, you have to take whatever magnetization has grown back, excite it again to get your next batch of signal. And you can't wait an infinite time, so things don't grow back fully. Okay, so it turns out that if you have tissue that has a longer T1 property, i.e. it takes longer for that magnetization to recover, you have less signal the next time you excite. So it means you detect a lower signal successively. So this is T1 weighted imaging. So longer T1s, darker tissue. The most common application that I think most people see here, especially if you image with us in our systems for fMRI, is in MP-RAGE images. So this is a typical MP-RAGE. The white matter here is actually nice and bright. So that means that it should have a shorter T1 than the gray matter. This tissue in this contrast weighting is also gray. So this here matches up. Okay. The next, very basic type of contrast is what we call T2. Where that comes from is the fact that the magnetization we're detecting turns out to be an aggregate of all the protons in your pool that you've excited and are detecting. So remember, all of these are atomic species, nuclear species. So the magnetization that they have, I think, is very, very minuscule. But in aggregate, we can see it. And in aggregate, everybody kind of has to be aligned for us to detect them, for the aggregate to be bigger. It turns out that as things evolve over time and everything is in a little bit different environment, they tend to fall out of step with each other. And then the coherence dies away. And then your signal you're detecting decays away. So that's what we refer to as T2 decay. So that's depicted here, where the yellow arrow kind of supposed to represent the sum of all the smaller red arrows. And you can see as those lose coherence with each other, that net sum gets smaller. So this here says T2 contrast or T2 decay. But the converse here is that if you have a longer T2, you have brighter signal because your signal hangs around longer that you're detecting. Okay. So this is what a T2 weighted image now looks like, where the gray matter has a longer T2, but in this case, it is brighter. So here, the white matter, the axons, the fibers, shorter T2s, so those decay away more quickly, so you have a darker signal here. And then what we do for bold is very much related to this slight variation. Where bold contrast comes in is it takes this idea of coherence of your signal, but now it looks at the decay with macroscopic or semi-macroscopic background gradients coming into play. So with natural T2, you're looking at maybe the difference between like the environment in one organelle in your cell versus another organelle. These are not like what we'd consider large in the grand scheme of things. But what we're looking at with bold here are kind of like the gradients inside of the blood vessels versus outside. So when Dr. Bannatyne talked about where bold contrast came from, before, if you're looking at, say, motor cortex of the brain, you're not actively engaged in a task, you have some deoxyhemoglobin in that region. And that deoxyhemoglobin is more paramagnetic, so it will tend to increase the field locally. So that's why inside the blood vessel here, which is what this circle represents, it's a cross-section of the blood vessel going through the tissue, the field lines are closer together. So that means the magnetic field is stronger inside the blood vessel. Outside of the tissue, outside of the blood vessel is tissue, and there it's more pure water cells, and that's more diamagnetic. Field is weaker there, field lines are further apart. So there is this field gradient between intravascular and extravascular. And this will cause, if you try to detect the MR signal in here, this will decay away much more quickly than what happens in this case. Now here, you have brain activation happening. So although you may have more utilization or energy usage, the system tends to oversupply blood, it tends to oversupply nutrients. So you have a net drop in deoxyhemoglobin or increase in oxyhemoglobin. So then that makes the inside of the blood vessels less paramagnetic, i.e. more diamagnetic, i.e. more like the outside tissues, so more uniform. So in a situation like this, where now the fields between the insides and outside are more uniform, you will have less dephasing in your T2 decay, so you'll have a stronger signal. And that's what you see if you look at an experimental time course in a voxel of brain tissue doing a task. The troughs in the red represent where you're not doing a task. And here now you have the equilibrium state where you have more deoxyhemoglobin, more dephasing, lower signal. You're doing the task, you have more blood flow, more oxygenation, but you have an oversupply, so you have less deoxyhemoglobin, less dephasing, more signal. And this is at the basis of these statistical maps that we compute for our work, and where we try to figure out, okay, what parts of the brain are active, connected, etc. Okay. And these are the couple of very basic types of contrasts that we use, that we leverage most. But MR, because of the sampling, the encoding, and the environments that we're looking at, there is actually a lot more that we don't do routinely, but is possible and available. So there is, flare, ionization, transfer, perfusion, fusion, and a few others I didn't get on here. But suffice it to say, there's a lot you can do with the physics, and you can get some very, very pretty images out of it. And hopefully useful. Okay. Any questions on? Okay. So I'll just have final couple slides here. So when you thinking about your experiments, we have, some things that you're hoping you need to keep in mind. So the trend in, our area has been to getting stronger and stronger magnetic fields. The primary reason for this is because with a stronger main field, you have a more, you can magnetize your sample, i.e. your subject or whatever you put in the system more strongly. That gives you more signal to work with. So there's no other easy way to do that other than building stronger magnet. So with this larger signal, you can choose to image faster, you can choose to image at a higher resolution. It gives you another degree of flexibility. Also, with this change in the fields and then how the our quantum or little microscopic magnets interact, you will have differences in T1 and T2 properties as you change fields. So that could be another thing that you exploit. However, with higher fields, remember omega 0 is equal to gamma B0. So as your B0 increases, so does your omega 0. So that means that you will be using more energy. You will be putting more energy into your sample, into your subject. So especially for subjects with implants, these are some things to keep in mind. And as the 70 fields are becoming more common in the clinic, and here folks have been working through these questions and trying to come up with reasonable answers. I think we've also finally been getting a better handle now on, as these fields get larger, it was more of a challenge to keep the main magnetic field and the RF fields more uniform, but those are now becoming a little bit more routine. And I think the final thing to just keep in mind here is that you may not always want to have a bigger magnet, a stronger field. The Cardiac Institute recently spent a lot of energy and a lot of resources on trying to develop a lower field system for the work that they were doing and supporting, with some really, really cool applications and results. So not everything is solved by going to higher fields, bigger magnets, stronger magnets. So it really is worth thinking about your problem and how you answer your questions. Again, you want to look at how your relaxation times, your T1, your T2s are varying with the fields that you are targeting. Again, with covering K-space, getting things at high enough resolution to answer your questions. This is the eternal trade-off with us hopefully getting our hands on local gradients soon. We'll have more leverage to not give up as much, but fundamentally that will not completely disappear. One aspect of image processing and reconstruction I didn't even talk about here was using acceleration and this is where, with how we encode data in Fourier space, there are techniques that let you skip some of that data, but then use information about geometries that let you fill that information back in. There are signal to noise trade-offs and other penalties. So there's, again, careful thought about how you make those trade-offs. And then the final thing that folks should think about, what other data you want to collect, that you need to collect to answer the questions that you're asking. One thing that our facility has become very good at and set up to do is we have a lot of standard additional equipment to collect eye tracking data. You can collect physiology data. You can even collect other types of MR data, but that help with your post-processing and your downstream pipeline analyses. So if there is, if you have concerns about breathing, distortion, if it's worth conversations with ourselves, the AFNI team, and seeing, how you set up and optimize your acquisitions to have the information on hand that you need. All right, that is all I had. Call it and take any questions.